All right, thank you everyone for showing up for the seminar today, and welcome to the Andlinger Center for Energy and the Environment. This is a highlight seminar to be given by Gabriella Schlau-Cohen. Before I introduce Gabriella, let me just mention that we also have folks joining us virtually online. For those of you joining us virtually, please feel free to enter questions into the Q&A box or chat, whatever it happens to be. And we'll answer those questions at the conclusion of the presentation. For those of us attending here in person, please wait for the handheld microphone to be passed around to be able to ask your question, because that will enable those joining us virtually to be able to hear it. So Gabriella is an associate professor in the Department of Chemistry at MIT. She has her BS in chemical physics from Brown and a PhD in physical chemistry from Berkeley. She's the recipient of a number of awards: the AAAS Marion Milligan Mason Award, the ACS Pure Chemistry Award. I like that, pure chemistry. And the Biophysical Society's Margaret Oakley Dayhoff Award. In addition, she is the co-director of BioLEC, of BioLEC, which is a Department of Energy EFRC, or Energy Frontier Research Center. BioLEC stands for Bioinspired Light-Escalated Chemistry. Its principal focus is on solar photochemical processes, and among her other research, she's going to tell us about that. Gabriella, please.
All right, well, thank you, Barry, for that introduction. And also thanks to the Andlinger Center for the invitation to come and visit you today. It's actually my first in-person seminar since the pandemic. So it's a nice sign of kind of return of these kinds of interactions. So like Barry mentioned, I'm here to tell you today about BioLEC, which is an Energy Frontier Research Center funded by the Department of Energy and also supported by Princeton, including the Andlinger Center, and we're really grateful for that support. So what BioLEC does is we bring together research groups from around the country to think about new ways that we can use the power of light for energy efficient or energy relevant chemistry. And so we're motivated to do this in part by the statistics shown here. So chemical manufacturing is an enormous industry. It's responsible for 1/4 of the world's GDP. And along with that enormous scope comes enormous consumption. Chemical manufacturing uses 10% of global energy, and in the process generates 7% of the world's greenhouse gases. Now, this is only on track to increase. In fact, the usage of fossil fuels for chemical manufacturing is going to exceed even that of transportation by 2050. Now, in part, this is because other industries, like transportation, have developed sustainable energy sources. And that means there's an urgent need for that kind of innovation in chemical manufacturing. So if we think about the problem, the fundamental issue is that, showing up on my screen, well, whatever. The fundamental issue is that in traditional chemical manufacturing, fossil fuels are used to generate both the raw materials as well as to achieve the high temperatures and high pressures required for reactions to proceed. But what we aim to do in BioLEC is something different. We want to use sustainable raw materials like biomass or waste products, and then use the power of light to make the reaction proceed. And so what that means is the mission of BioLEC is to use light to enable the directed editing to generate complex molecules for energy relevant applications. And so the grand challenge associated with that mission is that in order to make those molecules, we need to generate or break bonds that in a process that requires energy much greater than thermal energy that available in ground state reactions. And so for us, what success would look like is finding new ways to think about how to tackle those challenging reactions. So we focus on things like new fundamental chemistry, like phenol activation, or new ways of degrading environmentally harmful products, or upcycling some of the byproducts of plastic usage. And so this is what we want to achieve. Now, how we do this is we turn to nature for inspiration. And in particular, photosynthesis converts 200 billion tons of CO2 to biomass each year using just the power of light. So we want to look at that process, think about how we can take lessons from how nature works and apply them in chemical reactions. And so in order to do this, we target many aspects of the reaction. And I'll talk about how we do this using this very traditional picture in chemistry, where we have a generalized reaction coordinate and energy. And what we can see here is that there's reactants and products. But the kinds of energy that the kinds of reactions that BioLEC is interested in are ones that are uphill, where the products are at higher energy than the reactants. And in order to power that uphill reaction, we use photocatalysts. And so we want to improve on all aspects of this energy landscape. So in order to do that, we work in these interdisciplinary and interrelated aspects by developing new fundamental transition metal catalysts. We also think about ways we can, in particular, use biological building blocks or take inspiration from biology to improve the kinetics, efficiency, and specificity of how that reaction occurs. We also think about the fundamental photophysics or the energetics of our catalysts, as well as the reaction, to think about how we can generate even higher energy products, particularly for solar applications. And there's also work in the center that develops the reactions themselves. Think about transformations that will generate valuable products. And so in order to tackle all this, we take a very interdisciplinary approach involving PIs from multiple arenas. So shown here is some of the expertise in organic synthesis, including 2 groups here in Princeton, as well as groups external to the university. There's also groups in inorganic synthesis that think about developing, in particular, new earth-abundant catalysts. There's expertise in biochemistry, my group, as well as the Heister Lab, where that allows us to co-opt biological building blocks or learn more about the biological systems themselves. We also have expertise in photochemistry and photophysics to think about what are the relevant processes for these excited state dynamics. And then there's expertise in materials science. We can think about taking these lessons and incorporating them even into devices. So all of these areas are traditional areas within the chemical sciences. But although they are, broadly speaking, within the chemical sciences, they have traditionally operated in a really siloed way. And so one of the overarching principles by which BioLEC has succeeded is bringing together these typically siloed disciplines. And I forgot the spectroscopy, which is kind of ironic because that's the heart of what I do. But we and many others use spectroscopy to really build that mechanistic understanding to guide the next step of the development. Okay, and so what does this collaboration really look like in the EFRC? This is kind of a fundamental aspect of the motivation to even have a center. So all of the PIs within the center work in collaborative projects with multiple other PIs. And this is a network map of all of these interactions. And you can see that this collaboration really spans the center. And this approach has been enormously successful. BioLEC is only 4 years old. And even in the first 4 years, we've published 42 papers, many in high impact journals, and recently received notification that this success was recognized with almost 13 million in renewed funding that they promise is going to arrive any day now. All right. So so that renewal and that success was built on a lot of scientific advances. So I'm going to tell you about a couple just different snapshots of different scientific successes. So one real focus of the center is about building new tools that allow us to understand the function of the catalyst. This is a collaboration with the Rumpel's group at NRAL, along with the Knowles and Scholes group here at Princeton, where time-resolved dielectric loss spectroscopy was repurposed to look at these iridium catalysts. And what that combination of tools allowed these groups to do was identify the role of the counterion in introducing reactivity. So the counterion is always a part of the reaction mixture, but its role had never before been fully understood, which meant that it was an underutilized parameter in trying to optimize the reactions. So along with this building of new tools, there's also a lot of emphasis in the center of developing new catalysts. For example, this is a collaboration with Dave McMillan here at Princeton, as well as Matt Bird at Brookhaven National Lab, where Matt used pulsed radiolysis to look at the generation of productive intermediates in a cross-coupling reaction. And so that application of the new tools really guided the development of catalysts that led to those intermediates to improve their reactive properties. And so in both of these examples, a powerful aspect of BioLEC is the combination of research in the academic setting with that at the national labs and using a lot of the tools that are available at the national labs. And then finally, as we develop tools to understand the catalysts, develop better catalysts themselves, we also think about where this can be applied. So here's an example from Rob Knowles' group, also here at Princeton, where he developed a new light-driven reaction to depolymerize different polymers into their monomer byproducts for more environmentally efficient degradation and then reuse. So those are some snapshots of the kind of work that spans the center. So now I'm going to switch gears and for the rest of my talk, tell you about an area of research led by my group that spans many different groups, I think 5 different groups within the center. And so what we're all coming together to work on is to think about ways we can introduce dedicated energy capture systems so that we have light harvesting to increase the flexibility, the functionality, and the capacity of these photocatalysts. And so the specific type of photocatalysis that we started with, and it's the focus of much of the center, is called photoredox catalysis, pioneered by Dave McMillan here at Princeton. And so what this field does is use small molecule catalysts like this, ruthenium tris-BP shown here, as a photocatalyst. It's already widely used in organic chemistry. It's been demonstrated for a host of applications, including pharmaceuticals, chemical feedstocks, biomedicine, and polymers. So we want to think about lifting it to the next level for these energy relevant applications. And in particular, one challenge in the implementation of photoredox catalysis is they are often light limited. So if we think about light and the electromagnetic spectrum, typically photoredox catalysts absorb in this region here, in the 400 to 500 nanometer range. Now this region overlaps with substrate absorption, so it can generate unwanted chemistry. And it also, this whole region is highly phototoxic to biological materials, limiting its applicability for biological manipulations. And so ideally, we would have absorption here, kind of at the maximum of sunlight that allows us to do the energy capture in a way that's orthogonal to a lot of the other activity of our reaction mixture. But if we think about a molecule like this one, it doesn't absorb in that region, and it also has an extremely low molar absorptivity. We can rationalize that by thinking about this function. So what this photocatalyst does, it absorbs light and then undergoes a charge transfer event. And its absorbing states have a lot of charge transfer character. So just like many other charge transfer states, they have low molar absorptivity because you don't have that stable redistribution of electron density that you have with the strong absorber. And so it's because of all these challenges that really the light absorption, that energy capture properties are not yet optimized in photoredox catalysis. And so if we want to think about tackling this problem, we can again turn to nature for inspiration. So if you look at that leaf and you zoom in, what you see is you see dedicated protein machinery called reaction centers that are responsible for driving charge separation. So they have rapid electron transfer that then generates a stable charge separated state to perform high energy chemistry. Now, I'll just note that in another project in the center that I don't have time to talk about, we also mimic how they generate that stable temporary storage of electrons in photoredox catalysis, and that's work done in part by Rob Knowles here at Princeton, so you should ask him about that project. But anyways, so there's these reaction centers, right, that are responsible for this charge separation. But if we look at these proteins, in photosynthesis, they're surrounded by another set of proteins called the antenna. These bind chlorophyll, so they have high molar absorptivity and a broad spectral range. So they perform the dedicated energy capture. So they absorb the sunlight and then transfer that energy to the reaction centers. So unlike photoredox, in nature, the active energy capture and reactivity are separated into dedicated protein machineries. And so what we wanted to think about is if we can mimic that architecture, where we can attach an external light harvester to capture light and then transfer that light energy to the catalyst. And so we wanted to do this with energy transfer because energy transfer is a far field effect. It has a 10 times longer distance dependence than other schemes to sensitize photocatalysts, which use electron transfer or triplet transfer. So we wanted to demonstrate this with energy transfer. And so we started with these building blocks. So we have this RuBiPi, which is the canonical photoredox catalyst. And we have a protein called R-phycoerythrin, or RPE, which binds pigments in high densities. This is the native light harvesting protein from red algae. So we took these two components and then conjugated them using the NHS ester to react with the amines on the protein to generate this biohybrid construct where RPE was decorated with 8 to 10 photoredox catalysts. If we want to understand the energy capture properties of our biohybrid, we can just start by looking at the linear absorption or UV-Vis spectrum. So that's shown right here with the 3 individual components. And what you can see is that the biohybrid absorption spectrum, shown in orange, is dominated by the RPE or R-phycoerythrin absorption. And so what this allows us to do is generate this biohybrid that has absorption across the visible. We've moved that absorption into our desired range up to around 600 nanometers. And the extinction coefficient is 100-fold greater than that of RuBiPi alone. So now we've taken care of that energy capture piece of it, but what happens to that energy? So to figure that out, we turn to time-resolved spectroscopy. And so these are the fluorescence lifetime decay traces for our biohybrid catalyst, shown in orange, and for RPE alone, shown in red. And so what we see is the excited state decay decreases significantly for the biohybrid catalyst, which is a canonical spectroscopic signature of energy transfer from RPE. And so what these lifetime values tell us is that there's around an 85% efficiency in the energy transfer from RPE to RuBiPi. And that agrees well with some theoretical calculations that we did that use Förster theory, a theory of energy transfer, which gave us a predicted value of around 78% efficiency. So this is a strong indication that we have energy transfer from RPE, But to investigate these excited state dynamics further, we turn to transient absorption spectroscopy. So this is ultrafast transient absorption spectroscopy of RPE. And so what we see for RPE alone is an average decay of the excited state of about 1.5 nanoseconds. If we look at the biohybrid, we see a significant shortening of that lifetime, consistent with what we saw in the fluorescence-based measurements. And we see a decrease in the excited state lifetime by an order of magnitude, giving us an efficiency of around 90%, which is similar to the value that we got with the fluorescence lifetime measurements. So this is all strongly supporting this picture that we have energy transfer from RPE to RuBiPi. But to further confirm this, we can look at spectrally resolved transient absorption to look at the spectra of the excited state species. And so what we see here is the spectra of all three components. RPE, just with a mixture of RuBiPi, is shown in red, and we see just the bleach of RPE. Shown in blue is the excited state spectrum of RuBiPi. And shown in orange is the spectrum of the biohybrid conjugate upon excitation of RPE. And so what we see is when we excite RPE, we see in this region here the spectroscopic signatures of an excited RuBiPi, confirming that we have energy transfer from RPE to RuBiPi. So if we think about the overall goal of this project, what we wanted to do here is we wanted to enhance photocatalysis by using light harvesting. And so we were able to achieve efficient and broad energy capture and show that that energy we can successfully transfer to RuBiPi. But what about this overarching goal of improving catalysis? What is the impact of having light harvesting on catalysis? So to investigate that, we turn to reactions, and this is all in collaboration with Dave McMillan and Abby Doyle. And in particular, we looked at radical chain reactions, where your photoredox cycle generates an initiator that then reacts again and again in a chain-like manner to yield your desired product. And we looked at this class of reactions because they're particularly promising for industrial applications, because you're not limited to one-to-one stoichiometry. So it allows you to get to large scale quickly. And so we first looked at this Thylene coupling reaction, which is useful in late stage protein modification. For example, if you want to label a protein. And so we looked at the yields under, first of all, this is typically done under blue light, but we wanted to look at it under green light illumination. And what we saw is low yields with RuBiPi alone, some low level of activity with RPE alone, and synthetically viable yields of 73% with our biohybrid. If we look at red light, which has been a long-standing challenge in the field of photoredox catalysis, what we're able to see is it affords our product in relatively high yield, 30%, at least useful, but uniquely with our biohybrid. So what these series of reactions showed us is that the introduction of light harvesting enables or enhances product yields by an order of magnitude or more. We then looked at the scope of this reaction by looking at many different substrates. And what we saw is that we were able to reproduce this performance with a broad substrate scope. It's illustrated here. And so again, this substrate scope showed that we could enable or enhance product yields using our biohybrid. To further demonstrate the versatility of our biohybrid, we turn to a similar radical chain reaction, a cysteine desulfurization, shown here, which is typically used to couple different peptides together. And so what we saw was even better performance of our biohybrid in this reaction case, where we were able to achieve 100% product yields with our biohybrid, and no detectable yields were present for RuBiPi or RPE at green or red light. Another exciting aspect about our biohybrid is that we can easily purify it from our products or excess substrate, and then use that biohybrid again and again. Typically, you have to synthesize a new catalyst for every time you want to run a reaction, which can be a really expensive part of doing this chemistry. But because there's a huge protein attached, we can easily purify it out and then reuse it. And we saw around 80% yields with reuse of the original reaction. So this tells us when we think about enhancing photocatalysis with light harvesting, not only are we able to achieve efficient and broad energy transfer, we're able to use that light, even red light, to afford products in high yields. All right, so this is a proof of principle demonstration that this kind of architecture that we see in nature can be applied in organic synthesis. But can we generalize this strategy? Are there other cases where we can have similar success? So to think about this, we thought about this in the context of enzymes. Now, enzymatic reactions are particularly powerful because you're able to get a desired product with high stereoselectivity. So within the active site of an enzyme, they constrain the substrate in a geometry that yields your desired enantiomer. And so we looked at this ene reductase enzyme, which is the Heister group has demonstrated can have non-natural reactivity for chemistry, particularly with pharmaceutical relevant applications. We could think about building on to generalize for energy relevant applications. And this enzyme, similar to the photoredox catalyst I talked about in the first half, is often limited by light, because you have this charge transfer complex that is absorbing that for all those same reasons has a low extinction coefficient. And so we took this photoenzyme and this suite of commercially available dyes that are bright and photostable and conjugated them using similar chemistry to what we used in the previous example, where in this case, we have one enzyme decorated with around 5 to 6 different absorbers. So similar to what I told you about before, we now have a different bioconjugate with a light harvesting and a reactive component. And to characterize its energy capture properties, we can again go to the UV-Vis or linear absorption spectrum. And once again, we see a tiny bit of absorption, this low charge transfer state absorption from the photoenzyme, and broad and significant absorption from the dyes. Now, one thing that's useful about the introduction of this dye-based strategy is we can easily tune the absorption maximum depending on our desired application or our desired situation, potentially even introducing the possibility of multiplexing for multi-catalytic reactions. And along with that tunability, what we see is a 10-fold plus increase in the extinction coefficient. So again, we've been able to demonstrate that there is efficient energy capture, but we wanted to again turn to time-resolved spectroscopy to see what happens to that energy. And so here are the fluorescence lifetime decay traces for 3 different conjugates that I was showing. And we can compare the conjugate to a labeled non-photoactive protein. And what we see is in all cases a significant decrease in lifetime, corresponding to an energy transfer efficiency up to around 80%. And the values that we got experimentally are primarily consistent with those predicted from energy transfer calculations. In this case, the experiment was lower, we think due to some inefficiencies or non-optimization in terms of the labeling. But then with this efficient energy transfer and efficient light harvesting within these conjugates, we wanted to again go back to the chemistry to look at the performance. And so we evaluated the performance in these lactam cyclization reactions that the Heister group had previously disclosed. And so we used a green LED in this case, unlike the cyan LED used previously, with an intensity 3 orders of magnitude lower than that's what's typically used. Typically, you have a really high power LED that leads to hot reactions, can generate all sorts of unwanted effects. And so here we're much, much lower. And if we look at the product yield as a function of power in this microwatt regime as opposed to the milliwatt regime used previously, what we see is that the conjugate has a 5-fold increase in product yields. And that's basically consistent for all powers. Another thing that we saw about that improved with the introduction of light harvesting is actually our enantiomeric ratio improved from 92.8 to 96.4. And we think this small but significant improvement is due to the increased specificity upon photoexcitation, because you're essentially controlling better how you funnel light to the reactive site. And again, to demonstrate a broader scope of this, we also looked at intermolecular hydroalkylation. And so what we saw here is that there was actually no reactivity with the photoenzyme alone, where we were able to get relatively high yields with our conjugate. So together, this is an example where we see the introduction of light harvesting not just improved, but actually again enabled product yields. And so in thinking about the goal of this project to enhance photocatalysis with light harvesting, we've been able to introduce efficient and broad energy capture. We've been able to use that energy in photocatalysis to get products in high yields with lower powers and lower photon energies. And we've been able to demonstrate that this is generalizable to multiple photocatalysts. All right, so with this, just want to emphasize that this is an example of the kind of success that we've had to date in the center. But as we think about building and continuing to grow the center, we've brought on a lot of new expertise because we want to take these tools developed in this first funding cycle and push them to the new level. So we have X-ray spectroscopy to look at the basically to look in more mechanistic detail at the reactions, some theoretical support, some additional spectroscopic expertise, as well as a former postdoc within the center who's now taken on a faculty position, is going to help think about designing the next generation of catalysts. And so in conclusion, what the organization and examples that I've told you about today show that we've been able to bring together wide-ranging expertise, groups in different areas that don't normally work together, to think about how we can understand nature and take that understanding and apply it to chemical synthesis. In particular, we've introduced bespoke energy capture systems. We've identified aspects of how photocatalysts function, introducing new parameters for their optimization. We've actually developed some new energy relevant reactions, like for polymer upcycling. And we've introduced molecular electron storage that hopefully will increase the stability and the capacity of some of these photocatalysts. And so as we look forward to the funding cycle, this next funding cycle, we want to take this expertise that has existed within the center and that we're bringing in to think about how we can translate these advances to more earth-abundant catalysts, as well as target even higher energy reactions, such as multi-electron or multi-photon effects. All right, so with that, I want to just take this last minute to thank everyone in the center. The center is really a team effort with an incredible and large team that spans multiple institutions shown here. And in particular, I want to thank the groups involved in the science that I talked about today, bringing light harvesting into the photocatalytic systems. So the first project that I talked about, attaching a photosynthetic protein to a photoredox catalyst, was led by a really phenomenal graduate student in my group, Paul Cesana, right here. And that was in close collaboration with multiple groups. The reactions and the demonstration of the reactions was led by a talented Princeton graduate student, Beryl Lee, in Dave McMillan's group, also in collaboration with Abby Doyle. Some of the spectrally resolved transient absorption with a nanosecond time delays was done by Sam Shepard in the lab of Phil Castellano at NC State. The second story that I told you about, thinking about the use of photoenzymes, was again led by Paul, this time in collaboration with Claire Page in the Heister lab. And with that, I am happy to answer your questions.
Can you use this photoradar stat-based technology in nanotherapeutics?
Yeah, so it's already been demonstrated in wound. You mean in like bio?
Right.
It's been demonstrated in wound healing, so it's certainly possible. But the demonstration that has been done to date is in wound healing, where you basically generate a polymer network over the wound to hold it closed. And the idea is if we have some of that power from light, it can be easily deployed in the field for example, for DoD type applications. I would also say that a huge push of the introduction of light harvesting is also to make it amenable to biological systems. Because if you shine a cell with light at 500 nanometers, you're going to have immediate phototoxicity, especially at high powers. But if you're able to use low powers at 600 nanometers, that makes a real difference for cell survival. So that's I think, an important step towards its application in biological systems.
Thank you.
Two questions back here.
Thank you. Hey, I have a question about the RPE. That's really high energy uphill transfer, right? Like if you look where it absorbs to where it actually the RPE absorbs, how do you sort of describe that?
Fret. So basically they're really close.
Yeah.
So, just using the spectra and doing a typical Fret calculation, that's how we got these values. So I think one of the things that people don't often appreciate with Fret is how much that distance parameter is the dominating parameter because it has R to the 6 scaling. So even if you have, say, less overlap, like even, divide by 3, a third overlap or something, that won't matter nearly as much as the distance. And that's why actually in the example of the dyes, although I know you asked about the RPE-RU example, you saw similar predicted energy transfer efficiencies for all those different spectra. Because what really dominated the efficiency was the fact that our linker length is really short, so everything's really close together.
And then,
and then I'll also say another advantage in that particular example is that you have multiple donors. So you have 8 to 10. So multiple acceptors. So in a Fret system where you have one donor to multiple acceptors, you have first order kinetics, and then you just sum all those rates to get the total efficiency.
so then so it transfers energy, the antenna transfers energy to RU. Why don't you see that emit? Like, why don't you see that bleach in the long time scale? Or do you see it in a long time scale, like nanoseconds?
Yeah, so the spectrally resolved, so unfortunately our setup is not designed to do the 400 nanometer, to get the 400 nanometer window, essentially because of our chirp mirror compression system.
Okay.
And so we did that in collaboration with Phil Castellano, who has this nanosecond TA. This is basically a prompt spectrum, he calls it, where you just like generate the excited state with nanosecond long pulses. So you're just identifying what species are there rather than being able to get at kinetics.
Got you. So, okay. So it just might send the energy back to the system, right?
Yeah, so there's probably not that much back transfer because you have this rapid intersystem crossing. So in some ways, it's very similar to what you see in photosynthesis, where you generate you generate that, you have that initial charge transfer reaction, and then you have this cascade, which takes the electron far away, farther than you can have back. So you have to have that next fast kinetic step to kind of increase the gap so that you don't have the back transfer.
But I was always, because like Rubipi is one of those weird systems where it has a lot of singlet character where it can still fret from right? McMillan showed in some of his papers, you can still fret from that like pseudo triplet state, right?
Yeah, you can still fret from the pseudo triplet state, but as you go there, you have a decrease in the oscillator strength, so you have a much lower probability.
Thank you very much.
Yeah.
I'm not original. Dan already asked all of my questions, so I'll just go ahead and pass it back.
I had the very same question as well. So while the microphone, Sarah, if you can bring it over there. Oh, there's another one. My question is in the same realm. The spectra you were showing, the steady state spectra, those were all absorption, right?
Yeah.
Okay, so then the FRET, what matters will be the emission of those
That's right.
donors. That should be even more. So I have the same question as Dan because I'm amazed that you have such a high energy uphill transfer. What you're looking at
Yeah.
in the transient is ground state bleach, right? It's not any excited state spectrum.
Well, we're showing that we have bleached the ground state of the rubipi, right? So for
But you don't know
RPE,
if that's,
so for RPE, you have both. You have stimulated emission and ground state bleach, right? For rubipi, you have a much lower stimulated emission. So all you see is you excite the RPE and then you've removed the absorption of the ground state bleach. And we know that that is populated through energy transfer. I didn't know this, but Phil told me, as opposed to electron transfer, because you don't see the anion signature that you would expect in that region.
okay, yeah, because I was gonna follow up with, how do you know it's not charge transfer?
Yeah.
Okay.
At these conjugation densities, do you expect the photoactive species to like interact with one another, like self-quench or anything like that?
No, they're far enough apart that we don't see any signatures of that. We wouldn't expect it.
Okay, we have one question online from Sijia Dong asking, how did you decide where to put the dyes in the enzyme?
So just where there were lysines. So we just, the NHS ester reacts with amines, so it just reacts with the lysines. Yeah. Please.
One thing that drove me nuts with the heists is they always had to purify everything, right? So have you done your conjugation in the lysate? Like, does it work in lysate?
It probably would, but we haven't done it. I mean, in other very
Yeah.
very different applications, we have sometimes we just do the conjugation in the lysate to decrease the number of purification steps.
Yeah.
But we have not demonstrated that.
Yeah, because then it becomes industry relevant because it's like,
Yeah,
Yeah.
it's also possible, if we did that, I would want to use a cheaper dye.
yeah.
Paul started off with something that has really well-behaved photophysics, right? But there are other cheaper dyes that, because it's going to react with all of
Everything.
with all the lysines, right? So
Yeah, but that's really interesting that it works in light. That's awesome.
another thing that you can do is you can label on the column. So you can do it in one step where you just label in the column.
This tagging column.
Yeah, so you just leave it before you do the wash, and you just incubate with the dye in the solution. And so that's another way to decrease the number of steps that might not have quite that same challenge of labeling everything.
Awesome. Thank you.
Last question. Sarah, Charlie.
It's really cool stuff, and I really like the idea of the multiplex reactions with the dyes. Different dyes attached and potentially, sequential reactions that you could kick off like that. So I'm wondering, just in terms of the scope of the future chemistry and what you can do with this, first to understand this, these are all homogeneous solution reactions that you're running right now.
That's right.
Okay, so it's diffusion control between the excited state and whatever the reactants are.
Yeah.
Okay, are there opportunities to do this in heterogeneous systems with surfaces? And in terms of product separations at the ends, that's all right now driven by some kind of chromatography separation stage, or what thinking of engineering next steps for this and where it goes.
Yeah, I think kind of the nature of the catalyst is you form a complex in solution. So you could imagine some local confinement. I don't know that a heterogeneous catalyst might not have the specificity that we wanted, so we might not go to that. But how, to my knowledge, though we don't push this area, how this has been applied in more industry relevant and large scale is by doing photo redox in flow reactors. So rather than confining to the surface, just do everything in flow. And that's where the field is kind of going to try and scale up.
Okay, let's thank Gabriella Schlau-Cohen again. Thank you.